Yep.
Okay, perfect. So I'm not an MDH person. I am a POP person. So POP stands for poly-ADP ribose polymerase. And it's where the topic of my research in the Department of Biochemistry at the University of Vermont. And last year I applied to this program and because I was very interested in developing my teaching skills. And I realized that there is a link between malate dehydrogenase and the protein that I'm studying. And that was very surprising because POP is in fact a family, a protein family that is mostly involved in genome stability. So here is a family, the POP proteins, and in human we have 17 POP. And most of the time people know POP1, but there are a lot of different proteins. And what is very interesting with these proteins is some have an enzymatic activity and some have not. And it's due to a difference in the catalytic site that is represented in purple. And their localization is very different as well. You have some that are in the cytoplasm, some that are in the nucleus. And for some, like POP15, we don't know at all. So in my lab, I'm mostly working on POP1 and POP2. It's impossible to work on all of them, of course. And for the cure today, that I'm starting, I'm only focusing on POP1. And I know that it's indicated that POP1 is located in the nucleus, but in fact, we know now that it's also present in the cytoplasm and in the mitochondria, and people are starting looking at their role. And as I mentioned, you may have heard about POP1. Why? It's because there are inhibitors that have been FDA approved and also approved by Europe to treat cancer, especially, cancer that are mutated for BRCA, which is a DNA repair protein. So I'm talking about POP, but I'm still not talking about this modification. So here is a list of most of the post-translational modification that we can find. And ADP ribosylation is presented here, and it's a reversible reaction. And this reaction of ADP ribosylation is not only found in human, because your bacteria are also able to do ADP ribosylation using other enzymes than POP. So it's a pretty broad modification. But let's look at the ADP ribosylation mediated by this protein. So you have a target protein, and what POP will do is use NAD+ as a substrate and cleave this molecule here between in the covalent bond between the nicotinamide and the ADP ribose. And the covalent bond between the target protein will happen with this hydroxyl group of the ADP ribose. And what will happen is you will have a first addition of ADP ribose on the enzyme, and it's what we call a mono-ADP ribosylation. So it's a negatively charged modification because you have two phosphate groups. So sometimes it's compared to phosphorylation, but the consequences are very different. And the beauty of POP1 is its ability to add more ADP ribose. So what will happen is on this first modification, POP will use other NAD+, cleave this bond and add this ADP ribose on the first ADP ribose. And this polymer of ADP ribose can be composed of just a couple of ADP ribose or hundreds of ADP ribose. And this polymer can be linear or branches. And we know that in function of the presence of only one ADP ribose, several of them, or branch polymer, the signaling is very different. So in function of the size of the polymer and its structure, you have a different cellular meaning. So the usual question is which amino acid can be modified? So initially we thought that it was only these two amino acids, aspartic and glutamic. And with the technology improving, we have realized that several other amino acids can be modified. And serine is particularly interested because it can be ADP ribosylated when it's not modified, but also when it's phosphorylated. So you have an ADP ribose on the top of the phosphate group of the serine. And we again know that in function of the amino acid that is modified, the cellular meaning is different, which make everything very complicated. So the mass spec technology has been fantastic for the PARP field because it really helps us to figure out what are the proteins that are modified, as well as the residues. And we have this database there that is free. You can just go to this database called ADP RiboDB, where all the proteins are listed, as well as their residues. And you have the link to the paper that is describing this modification. There is one main issue with this database. It's the fact that these mass spec studies are mostly done in response to DNA damage, because PARP is very active in following DNA damage. So we have difficulties to figure out if this modification can also happen in absence of DNA damage. So that is one of the issues that we have right now. So let's go back to ADP ribose and MDH. These study, mass spec study, were done by several labs, and they really identify proteins in every compartment that can be ADP ribosylated. And Hope et al. In 2020 focused on metabolism, and they build, for example, these figures where they show all proteins that are within the mitochondria, and mostly in beta oxidation, TCA, and oxidative phosphorylation that are modified. So what is happening is you have the different name of the genes, and when you have this circle, it's the frequency of this protein were found ADP ribosylated. And here we have MDH1 and MDH2. And what they show is MDH1 is ADP ribosylated on two residues, so serine 241 and glutamic acid E329. MDH2 is also ADP ribosylated on two amino acids, E84 and serine 317. And we go back to this question about the serine: is it modified when it's phosphorylated already or not? And is it possible that there is a competition between phosphorylation and ADP ribosylation? And I'm saying that because these two residues has been found phosphorylated as well. So when we think about a project, that can be a project where you study the competition between two post-translational modification and the consequence on the function of the MDH or the activity of MDH. So what will happen at UVM with my students? The main goal that we have is to characterize the role of pyrylation on MDH structure and function. And so I'm starting the first year, and I am mostly letting my students studying what they want in a certain, with some limits still, because we cannot do everything. And there is also the fact that we don't know nothing about MDH and PARP in terms of pure enzymatic activity. So the first main question is ADP ribosylation affecting MDH activity? So the student will compare the activity of MDH and mutated MDH at the residue that I just mentioned earlier. Another possibility is we have an in vitro assay to poly-ADP ribosylate MDH, and then we can test the enzymatic activity. So in place of mutating the residue, we can look what is happening when the post-translational modification is there. This modification, as I mentioned, is pretty large, up to 200 units of ADP ribose. So it may modulate the dimerization or trimerization of MDH. So this is also something that students can look at. And because of all these proteins involved in the TCA cycle, for the example, that are modified by PARP, we can also look if there is some changes in the MDH interactome.
2-minute warning.
Okay, thanks. Finally, I mentioned that we are mostly studying PARP1, but you have seen the family that there are 17 PARP. And there is a prediction from mass spec that PARP1 may not be the only protein that is modifying MDH1 and MDH2, but another one is also PARP10. And PARP10 is adding only one ADP ribose. So another confirmation would be to compare or to use all the PARP and to check which one is modifying MDH. And then go back to the activity of MDH and see what are the consequences between mono-ADP ribosylation versus poly-ADP ribosylation. So there are a lot of questions that can be asked by the students and just by using in vitro assays. So I want just to thank my undergraduate student this year who really helped me to build this cure. So her name is Maddie. She just graduated, so I was so happy for her and so sad at the same time. And I would like to thank you for your attention. And this is an old electron micrograph of the polymer of ADP ribose because it's a beautiful post-translational medication. Thank you.